I want to take an opportunity to thank Nasha for coming up and giving an amazing perspective on how powerful our parents in this room are. So let's just give it up for her one more time. You've already heard me talk. I'll skip the intro and cut it down. I'm Dylan, and I'm going to give you a State of the Union of sorts on some of the science projects because, as Mike mentioned, we're coming on the heels of a science conference that had really exciting things going on. But as a background for me, I am a scientist. I went to Vanderbilt, so all you Tennessee folks out there, I look forward to connecting with you. I went to Vanderbilt. I got a PhD in cell biology. What that means is I went to school for way too long for a piece of paper where I learned a lot about something very, very small. And with that, I took that experience and I live in New York right now, and prior to this position, I worked in a lab as a scientist doing some of these experiments. But in a science talk, this is the background that's important for people to understand. But in a room like this is the background that's important for you to understand. I mentioned before, I'm a brother to someone with Dup15q, so this is my wife over here, and this is my brother Travis, who's got isodicentric Dup15q. He is the greatest mentor that I've ever had in my life. He's nonverbal, so he's never spoken a word, and I always tell people that he's the wisest person that I've ever spoken to. So I want you to keep that in mind, all you siblings. You guys are doing great, and take the opportunities to learn from your siblings as well. Here at the Dup15q Alliance, we start every board meeting by thinking back to our motto and what we're here for, and that is that we empower individuals living with Dup15q syndrome to reach their full potential by advancing breakthrough research and life-changing therapeutic treatments, supporting families affected by Dup15q, and promoting advocacy. I'm on the science side. I'm going to be talking mostly about point number one here, about advancing the science, but I want to point out that these three pillars are all equally essential and important. So even though I'm going to be talking a lot about science, I know personally that family support and patient advocacy is absolutely invaluable. So even though I'm focusing on one area, don't consider that all three aspects are very, very important. The Dup15q Alliance has a mission to support science, and the question is, where do they fit into the process of developing a therapeutic? I think that's what everyone's hoping for, is that there's going to be a therapeutic for Dup15q syndrome. The question is, where can the Alliance facilitate this process? To make a therapeutic, you start off with an idea, and this idea usually comes from a scientist in a lab, and the question is that idea real? Can we adjust it? And can we actually test it in the lab so we have evidence that that idea is something that could actually work? So then you get your scientists, they go into the lab, they do all the experiments, and there's a lot of arrows here because there's a lot of experiments that are very expensive and time intensive, and they build off of each other. You learn one thing, you move on to the next thing, and they stockpile on each other. And so it's a long and expensive process. We hear this a lot from our scientists, especially in this current funding climate, about how grants are so hard to get now, and the science is so important to move forward. But this is a big process to actually have the idea tested in the lab to get to a point where there is a drug. And having a drug or a therapeutic is an amazing thing, but there is still a barrier where that drug needs to make it into a clinic and actually be tested in a trial to make sure does it work in people. So the Alliance over time has invested in different ways, and you're going to hear over the course of the conference different ways that the Alliance has invested in ideas, invested in helping scientists do their work, and then ultimately we're working on getting drugs into the clinic so we can have clinical trials coming up. The Alliance first started funding research in a big way back in 2013. So this is me. This is me in 2014, actually. And the reason that I have myself up here without the beard and longer hair and looking a little goofy as a college kid is because I got involved with Dup15q research as an undergraduate. I went to the University of Mississippi. I studied chemistry. And during my freshman year, an article came out, a press release from the Dup15q Alliance saying, hey, we are partnering with this guy at Texas A&M, and we're going to make a mouse model for Dup15q. And I thought, that's amazing. We're going to make a mouse that's meant to study what my brother has been dealing with for the last however many years. And so I sent an email to the researcher, his name's Scott Dindo, and I asked him, like, hey, would it be cool if I just stopped by the lab? Like, I'm interested in science, I'm studying chemistry, but I don't have lab experience. I just want to know what you're doing to help people like my brother. I'll sit in the corner, I'll wash dishes, I don't care, I just want to be there and see what's going on. And he said, absolutely not. You are not going to sit in the corner, you're going to be on the bench doing experiments. And it was the greatest gift that I had, to be thrown in full force And it changed my perspective. I wanted to be a medical doctor, but the experience in that lab changed me to get to the point where I wanted to study research and understand how we can develop these things for Dup15q. I also want to point out, this is true for a lot of our researchers, the greatest experience I had was talking to researchers and grad students and postdocs who work long hours, they work nights, they work weekends. The work-life boundary has been blended in ways that you have no idea. Well, you do have ideas, but it's, the key difference is that a lot of these scientists aren't personally affected by Dup15q syndrome. They don't have family members, they don't know people, they get into it likely because they studied a gene in grad school, they thought it was interesting, they stuck around with it because the grant money was there. But research on Dup15q syndrome is really difficult. It's a really tough nut to crack, but they're there because they have the opportunity to come to these conferences, and we have scientists here. They meet the families and they realize, like, this is an uphill battle, but it's one I'm willing to take on, and I'm gonna throw myself full force into it. So that, as a college kid who was interested in science, changed my whole perspective because I saw researchers who were working tirelessly to study Dup15q syndrome who had no personal connection to it. I'm really fortunate that some of my work from that lab was included in a publication that went out, and this was kind of highlighting some of the findings we had from the first mouse model of Dup15q syndrome. And this paper came out in 2017, so the funding started in 2013, mouse was made in 2014, paper came out in a relatively short time in 2017, so you can already see the time delay between funding and research hitting the market, but this process is absolutely invaluable. And one thing I want to highlight is that this mouse model studied a gene called UBE3A. You'll hear a lot about it. We heard a lot about it at the science conference, and we think it's a gene that's really important in Dup15q syndrome. This was the first mouse model. We've got researchers here and at the conference before who have built better mouse models. We learned from mistakes or caveats from here, and things are continuing to improve. So every time you guys hear, we're making a mouse model, well, why? It's because we're learning from new genetics, we're learning from what we find in our other mice, and we're getting better at making something that actually looks closer to what we see in some of the symptoms for Dup15q syndrome. So I mentioned UBE3A. I'll give you guys a very brief intro to it because you'll hear it in other sessions, but what is UBE3A? UBE3A is this colorful guy over here. It's very complicated looking. It's a protein, and a protein is something in a cell that actually does a function. So UBE3A has a function where it looks around the cell and it looks at different things that are around there, and it says, is this trash? Does the cell need this anymore? Can we get rid of it? So UBE3A is gonna look around the cell and it's gonna say, oh, look, this should be marked as trash. So it says, oh, this is trash, and we're gonna put a tag on it. So it puts a tag, it's called ubiquitin, and it puts a tag on these things in the cell that need to be broken down. And what happens is the cell sees that tag, it throws it away. That's good. This is a regulated process in the cell. The cell is made to make things that do functions, and at some point the cell doesn't need it anymore, breaks it down, and then we can start the process and make whatever we need at that point in time. Problem with Dup15q syndrome, as you might be aware, is that we've got extra genetic material in the genome, which means extra UBE3A. If you've got too much UBE3A, you're probably marking a lot of extra stuff for trash. You might be breaking down and throwing out more than you want to. And in some cases, you may actually be breaking down things that are really important in the cell that you don't want to be throwing away. So a lot of our understanding and the therapies that you'll hear about throughout the conference are saying if we've got too much UBE3A, how do we bring that back down towards normal levels so that we don't have it running amok in the cell? Now, the Dup15q Alliance, as I mentioned, has been involved in research in a big way since 2013. And one of the main ways that they've done that is through academic grants. So you may have heard about Dream Grant opportunities that support our researchers in a big way. And Mike made a very good point earlier about how when we fund our researchers, we move research forward in a way where those researchers make really good findings, and all of a sudden the NIH becomes interested. We get other organizations who say, that's really valuable. We want to put our money behind it too. So academic grants are, I think, one of the key ways that we have funded research at the very foundation. Academic grants can fund a lot of different projects. There's a lot of high overview topics I'm going to cover here, but just some ideas of basic science research is understanding what is UBE3A? What does it do? What are these things that it's throwing out in the cell? These are the questions that are going to be really important going forward. Making models like animal models. These are the mice that I was talking about. We make them better. We don't just have mice, though. We've got worms that we're developing. We've got flies. We've got all these different animal models that are coming out that are answering different questions about Dup15q. We're taking some of the basic science questions that we have, and we're putting it into mouse and saying, is this true? Is this a way that we can target something in a person? We also have something called patient-derived stem cells. You may have heard about this on the news or anything like that, but these are really valuable. You can take a blood sample from a person with Dup15q syndrome, and you can take that blood and you can put it through a few different processes in the lab, and you can turn that into stem cells that have the same genetic makeup of the person that it came from. It's really valuable, but the key is that you can then take that stem cell and turn it into a neuron. All of a sudden, you've got a dish of neurons from Susie and Joe that are basically mimicking what's going on inside their brain, and we can put drugs on them, and we can test them and understand what's going on in a Dup15q cell. And getting that nitty-gritty understanding is really, really valuable because it builds on the research that we already have, and it supports research in other areas. One of the ways that it builds on other research are through these research studies, and it's a broad term, but I'm talking about the research studies that you guys can participate in. These are family volunteer studies that we've encouraged signups for, and hopefully you guys are participating. These can include things like biomarker discovery. Biomarkers are something inside a cell that tells you, or there's something that you can extract in the doctor's office that tells you what's going on in the body. You may be familiar with where you go to the doctor, you get a blood test, and the result's high. Okay, this result's high. Does that mean that I have something wrong? Is there something that we can do to treat it? Biomarker discovery takes a reverse approach and says, we know you've got Dup15q. Let's look in your blood and let's see if there's something different. And the question is, if there's something different, if we bring it back to normal, does that help the symptoms? Or if we help the symptoms, do we see that going back to normal? So we can try and tie those two together to really get a clear idea of what's going on the inside, because it's so hard to understand that. Survey data collection. You guys are tirelessly familiar with surveys, I'm sure, at this point. And we know that. We are very careful with the surveys that we ask you guys to fill out. And we ask you to fill them out because they're all valuable and important information. The parent perspective is something that is absolutely invaluable. And you guys know, you go to the clinicians, and depending on which clinic you see, we've got a lot of experts, but some of you may be going to clinics where you know more than your doctor about the condition. It's a hard spot to be in, but you guys are your own experts. And so we want to capture all the knowledge you guys have so we can understand what our community is actually dealing with. And natural history studies are something I'm going to go over tomorrow in a really big way, but all the work that Citizen Health and SOAR are doing to advance natural history studies and understanding what does the natural history of Dup15q syndrome look like? What does it look like when you're a toddler, infant, if you're a teenager, if you're an adult? How is it going to change depending on your genotype? Maybe if you're isodicentric or interstitial, things might be a little different. What are the things that we know, and what are the things that we can then inform going forward, like my kid's 12? What's he going to look like at 14? These are questions that families have, and natural history is a great way to understand how Dup15q progresses over time. The last part where the Dup15q Alliance has been playing a really meaningful role, and this is a big push that's happened recently, is through therapeutic partnerships. So hopefully you've seen the announcements that we partnered with Kecho and Quiver this year, and they're working on ASO gene technology, basically, to regulate UBE3A and Dup15q syndrome. Therapeutic partnerships are really important because so many of the medicines that you guys take are Band-Aids. They're for seizures, they're for behavior, they're for sleep, but they don't address the root cause of Dup15q. They just treat the symptoms. The therapeutic partnerships that we're making are looking to address the root cause of the disorder in the hopes that we can have a really meaningful impact on symptoms. So we've got our therapeutic partners, and the hope is that we'll have clinical trials coming up. I know at the last conference there was some talk of clinical trials, and our hope is that next conference we'll be able to talk more and more, and every conference here on out we'll talk about clinical trials you guys can participate in. These therapeutic partnerships are essential to getting to that point where that's going to be a reality. Our therapeutic testing in mouse models is also really important because if you have a therapy, you got to make sure it works. And the best way to tell it works is putting it in a mouse model that we've already developed with the Dup15q Alliance that recapitulates some of the things that we see. So you can already start to see some cross-connection between these. And another part would be therapeutic testing in stem cell models. Again, Susie or Joe, what do their neurons look like? They're a little different than what we'd see in a neurotypical individual. If we give them a drug in a dish, does it kind of regulate that? It's a very good question, and these are the important questions that we need to ask before we start to move into a clinical trial. Because we're not just going to put you guys through the ringer without an absolute confidence that something looks like it's going to work and it's going to be safe. This is all essential work that needs to happen before we can get to clinical trials because we value our families and we want you guys to have the best experiences possible. And what I hope you've recognized now is that these do kind of overlap and they intertwine, and what we learn from some data sets can inform others, and they all go back and forth. It's this really kind of nebulous combination of scientific experience that manifests from our clinics, from our pharma partners, and our academic researchers all working together. And at the science conferences, it was really great to see all of them working together in the same room, and it's something so powerful. But for now, I'm just going to talk about academic grants as the first introduction to give you guys an idea of what's going on right now in the sphere of academic grant work. Mike highlighted these numbers. We had an old number where for every $1 of alliance-funded research that we supported through a grant, researchers had $6 of additional funding. And that 6x multiplier is amazing. That's a huge number when it comes to research. We've updated it up to 10. So we are now, we're 10xing every dollar that goes into research. And some of those grants I'm going to highlight on this next slide here, where we will support research at the very basic stage. Let's try and make a mouse model. You guys find something cool in a mouse model, take that to the NIH. NIH gives you a couple $100,000 to study that in a really meaningful way. So here I'm highlighting 4 research grants that came around in the last 12 months that are studying specifically Dup15q syndrome. The first one I'm going to talk about, and I'll have him stand up a minute, is Dr. Matt Judson. He works at UNC, and he is working on testing in one of our therapeutic partner, their molecules actually in a mouse model of Dup15q. So Matt, would you be able to stand up a bit so everyone can acknowledge you? Matt's been with us for a long, long time working on Dup15q. He's on the big give every year, I feel like, talking about his work, and he's going to be on some panels and sessions later. So find him and talk to him and ask him about his work because it's really, really cool. We also have a researcher here who was funded through the $1 Million Bike Ride. This is a partnership we have with the University of Pennsylvania. They raise money through a bike ride, and then they give it out to a bunch of different researchers who study rare diseases. So Dr. Alonzo Slaver is actually in Israel, and we were surprised by this. We'd never heard of him. He was a new researcher who learned about Dup15q, presumably through some of the stuff we were doing at the Alliance, and said, that's something I want to study. I want to work on making a model that can study Dup15q in a way that we haven't before. So he actually uses worms. Worms are a really interesting model to use in the lab, and you think it's probably kind of weird as a parent, because as a scientist, I thought it was weird too. But one of the cool things you can do is you can play with the genes, and in a mouse, it's really easy to take one, not easy. It's more straightforward to turn one gene up or turn one gene down. In a worm, you can do all sorts of combinations. And we know in Dup15q, there's tons and tons of genes that are involved, and UB3A seems like a really important one, but we don't really know or have a great grasp of what all those other genes are doing. And we know from our parents, like, if you've got a big duplication or a small duplication, things can look a little different. So we want to understand, what are those other genes doing? It's a very open question. And so he's actually entering our research field so that he can answer some of those questions with all of the knowledge that he has already. We also have 2 grants that were funded by the Eagles Autism Foundation. This is a partnership with the Philadelphia Eagles, where they sponsor research that's broadly into autism in general. They don't have a bias towards Dup15q at all. They just fund autism research, which is fantastic. But both of these researchers have originally been sponsored and supported by the Alliance in one way or another. And now, with the findings that they were able to generate with Alliance-backed research dollars, they can go on to get extra money from these other sources. So these 2 researchers got really big grants from the Philadelphia Eagles, which I grew up in Jersey, but I grew up as a Jets fan. But now I got to root for the Eagles because they're supporting our researchers, and I hope you guys all do too. Dr. Eric Levine at UConn is studying other Dup15q genes besides UB3A. And Dr. Jason Yee in St. Louis is looking at novel therapeutics to regulate UB3A. So these guys are really, they're in our field. They've been here for a while now, and they're taking the next step and moving research forward in a really meaningful way. I mentioned that we had a science conference, and that might be an understatement. We had a packed science conference. Between full day Monday, full day Tuesday, and half day Wednesday, we had 33 speakers that came. I almost lost my voice by the end of it, but we made it through. I wanted to save it for you guys, and we had so many discussions looking at Dup15q and Angelman syndrome, because we share a lot of researchers, and understanding what can we learn from Angelman that we can apply to Dup15q, what are some of the advances that they're making, What are the ways that we can work together to advance things moving forward? So these researchers have been fantastic and amazing, and there's a lot of hope coming out of those meetings. We had, as Mike said, about 130 people in person. We had about another 70 on Zoom. So you think about 200 researchers, clinicians, bench scientists, pharma companies in the room talking together about Dup15q. That's an amazing advancement for the field of science, and the talks that we're hearing are researchers who have been doing Dup15q who are making progress. We're hearing from new researchers who are coming into the field, talking about how they're starting up and learning from the experts in that space. So it's a really nice area where all of this knowledge and learning can feed off of each other. And I've never felt more excitement coming out of a scientific meeting than I did yesterday. So I'm really, really encouraged. On the scientific front, I don't have the ability to cover all 33 topics that we did, but I'll highlight a few here. So we talked about mouse models. That's a really big thing you guys are probably familiar with. And how do we make better mouse models to recapitulate what we see in Dup15q? Learned about fly models. I told you we've got different model organisms that are interesting to study. So flies, we've got a great study looking at seizures and how we can treat them there. Learned about speech acquisition, learned about vision issues and different things like that in a clinical setting. Is what are patients actually experiencing? How can we measure it better so that when it comes to a trial, we actually can look at what change looks like? We talked about ASOs. ASOs I'll mention a little bit later in my talk, but this is one of the therapeutics that's coming out to treat Dup15q, hopefully in the future, and we're going to have our therapeutic partners talk as well. And then lastly, drug screening. You know, you guys are taking a lot of medications at this point to manage all the symptoms, and one of the questions is, how well do they work? Can we make them better? Are there things that are being used in other indications that could work in Dup15q? So there's a lot of exploration and understanding of how can we be really smart about the drugs that we're giving our kids to treat their symptoms. And what I have down here is a graph that I'm going to explain in a bit, but it's basically showing, and forgive me, I'm a scientist, I speak in graphs. So on the x-axis here, we've got time. So going from left to right, we've got time over the last 10 years. And on the y-axis, we're counting the number of publications that talk about Dup15q. You can see from 2015, when I was at my last conference in Orlando, hearing about the mouse model, we've had a steady uptick in research, publications that talk about Dup15q. And in 2020, something happened that just impacted a scientist's ability to go into the lab. And I don't know what that was, but for whatever reason, scientists couldn't study in the lab. When COVID happened, labs shut down. And so we had a lot of researchers that published everything that they had when they were at home because they didn't have a choice to go into the lab. And so we had a little bit of a downtick because of this slowdown. But you can see that rebound has happened in a really, really strong way. And we had our highest number of publications on Dup15q syndrome last year. And we're only halfway through the year, so don't let the small number fool you. We've got publications on the way. And so we hope to see this really fantastic increase in studies on Dup15q coming out, which helps our scientists. They learn from each other, and it puts us on the radar of new scientists who can also learn from them. The next thing I'll talk about are research studies. You guys have heard about a few of these, hopefully. We've been kind of blasting your emails and the Facebook groups, trying to encourage you to sign up for studies. There's a lot of cool stuff going on, and you should have a packet in your welcome guide that has the locations for all these, but as we mentioned earlier, fine staff members will point you to where you need to go. But I'll briefly cover some of the studies that we have. So SOAR and Citizen Health are two natural history studies that are kind of working in parallel, and they're very complementary. The question is, what does Dup15q syndrome look like over time? What does the spectrum of our disorder look like? And that's the way that they're going to capture it, is through these natural history studies. So SOAR is something that our researchers in Los Angeles are doing. They traveled here. They're going to be studying some kids here. And Citizen Health, we've already heard a little bit about how you can take electronic records and use those to advance our understanding of Dup15q syndrome. We also have a researcher here, Dr. Danny Miller, who's going to be here just for the early part of today. So if you're interested in this study, I'd recommend that you find him and participate. He's looking at collecting blood samples in a way that doesn't use a traditional needle and a vein. He's got something that's kind of like the passive sensors for diabetes that you've seen. And he can use something that is akin to that. It's a lot less painful or uncomfortable than a needle to collect blood. And he's looking at changes in the DNA of Dup15q. So he spoke at the research conference. I'm personally really excited about his work, so I hope that as many of you as possible can participate in his project. We also have Combined Brain here present. They're working on a study to understand, what are some of those biomarkers? What are the things going on in blood that could be indicating what's going on in Dup15q syndrome? They're really nice. They're They are a nonprofit, but they partner with so many rare disease groups because we're all going through the same questions of trying to answer, how can we get to the clinic and how can we help our families? And they're basically combining efforts so we're not being repetitive, and they have a nice connection to pharma. So pharma's actually reached out to them and said, hey, we're interested in Dup15q, but we need to learn more before we can fully commit. So Combined Brain gave us a nudge and said, hey, by the way, pharma's interested. If your families give blood at the conference, pharma's gonna get some valuable information. So we want you guys to consider participating in that trial as well, or that study. And the last one here, I'm highlighting Rajuda Wilson. She comes from UCLA, and she's performing a motor study. So hopefully some of you have signed up for it, but the idea there is that motor function is pretty wide in our community, and there's not a great study of how does motor symptoms affect other domains. Do motor difficulties perhaps interfere with kind of individual functioning when it comes to eating? Does it impact your ability to communicate with an adaptive communication device? These are questions that are outstanding in the community and one that she's going to take a really nice approach to study. And I mentioned natural history studies. I've got a talk tomorrow, so please tune in for that, and I will go very in-depth as to what those look like. The last thing I'll talk about in this arm are therapeutic partnerships. This is something that we're really excited about because in a meaningful way, the Dup15q Alliance has started investing in these companies that are developing therapeutics for Dup15q. So here I'm highlighting two individuals who are part of our community. I've got Sam Quigley over here on the left. He's a father to someone with Dup15q, and he will be speaking at other points, and I encourage you all to listen to his story. He started a company called Kicho that basically said, I'm sick of waiting for a therapeutic to come along. It's taking too long. I'm going to do this myself. So he started a company, and he's working to develop a therapeutic for Dup15q syndrome. The other individual here, Dr. James Fink, is a researcher who doesn't really have a connection to Dup15q. He's not personally affected by it, but as a grad student, he was in a researcher's lab who studied Dup15q, and the Alliance actually supported some of his grad school. I'll tell you what, I don't know if any of you went to grad school, but any extra dollar in grad school means everything. I'll give James a lot of credit. He dove in head first. So this was 10 years ago that the Alliance funded his research, and he has never quit. So he was in academic research. He got his PhD, earned that the hard way, and then he started working with a company that became Quiver. And at Quiver, he worked on therapeutic development and actually started and is leading a project studying a therapeutic for Dup15q. So both these gentlemen have been doing amazing work for a long, long time, and I really encourage you to listen to them and ask them questions about their journey and what they're doing. And we at the Alliance are really fortunate to have a hand in supporting their work in a really direct and meaningful way this year. The therapeutics that they're developing are things called ASOs, antisense oligonucleotides. You may have seen it in my email blasts. You may have seen it on social media. But just to get everyone a basic understanding, up here in your cell, you've got DNA. This is the chromosomes. This is all the genetic information that you have in the cell. But it's really, really precious. And the cell has to be very, very careful about it. So what a cell will do, it'll make something that looks like DNA. It's called mRNA. And it's got a nice sequence like DNA. So it's got all sorts of letters that specify how the cell can make something that does something in a cell. So a protein like UB3A, our colorful little ribbon friend down here, is made by the cell reading this string of letters on mRNA. The thing in a cell is if you have more mRNA, you get more protein. And we think that's what's going on. We've got extra DNA for UB3A, we get extra mRNA, we get extra protein. So the question is, at any point in this stage, can we bring that level down? Would that have an effect on the cell? And so one of the technologies that's come around, especially in the rare disease space, because it's so good with genetics, is something called an ASO, an antisense oligonucleotide. And it's got letters. And these letters, which are these little spikes here, are made to match up and marry up to exactly the mRNA that you want to target. So in this case, we've got an mRNA that will make UBE3A. We've got too much in the cell. What if we have a molecule that binds to it? And the cell recognizes this and says, OK, I see something wrong here. I'm going to throw that away. So we're not going to throw all of it away. We can adjust the levels. So instead of having too much, we can bring that level back down. So this is something that we've seen a lot of other rare disease groups succeed with. Angelman syndrome, which we work very closely with, has clinical trials ongoing for this type of technology targeting UBE3A. They want to bring it up to normal levels. We're bringing it down. But they've had so many successes in the clinic. And we're really encouraged by that it seems like a really viable strategy. And we're hoping that our therapeutic partners will ultimately make this a reality. So I want you to remember is that we've got these kind of three arms, if you will, of research that's ongoing, but at the center of it is the alliance. We've got these three pillars that are all equally important. We've got research, we've got advocacy, and we've got family support. But the alliance is really in the thick of all this research and facilitates a lot of these interactions happening so that our researchers can be ultimately as successful as possible, because the faster they figure stuff out, the faster you guys can benefit. The question that we're now wondering, and you guys are probably wondering, is what is the future, right? I gave you a lot of things to think about of the past and the ongoing, and you may be inundated, but what if I told you there's just as many things on our plates now and that are going to be coming up in the near future in all of these different avenues? So we've got academic grants that are going to be looking at improving our animal models. I mentioned that a little bit before. Mechanistic therapeutics is taking the idea of we're learning so much about UBE3A and how it basically throws things away in the cell. Can we learn about how it does that? Can we learn about what it's actually tagging to be broken down? And maybe then we can get an even more precise version of a therapeutic. We're looking at gene-specific contributions, so outside of UBE3A, what are some of these other genes doing? How do they contribute to Dup15q? And drug screens. Understanding maybe there's drugs that are already on the market, can we use them for Dup15q in one way or another, and testing these in models. In the clinical research side, we've got something genotype-phenotype correlations. This is a very science term, but the connection is genotype, interstitial isodicentric, big duplication, small duplication. Depending on what that looks like, what does your kid present with symptoms? We've got a general idea, but we don't got a really clear connection of if you've got these genotypes on your genetic report, what are the symptoms that come with that? So really trying to strengthen that correlation so we can help you guys understand a little better about what could be coming up as your kid develops. Improving clinical outcome measures is something that a lot of our research studies are doing, particularly that SOAR natural history study that Shefali and the team in Los Angeles is doing. Outcome measures are something when you look at a clinical trial, you want to know, did it work? Right? How do you measure that? I could ask you guys, you may have different things that you'd say you would measure, but ultimately it's the FDA that decides whether something works or not. So you have to have some sort of metric that says, meaningful change for our family is an improvement in communication, in seizure management, something like that, and how do you measure that? That's the key, you gotta be able to measure it. And so there's a lot of research of understanding, if we can figure out how to measure it now, when we get to a clinical trial, we're gonna have less road bumps or hurdles to navigate. And improving symptom management, you guys are so familiar with this. We wanna develop better ways that you guys can manage symptoms, whether it's through therapies or therapeutics or anything like that. Our therapeutic partnerships are a really exciting avenue that we're hoping to go forward with and expand. So we're looking at additional therapeutic modalities. I talked about ASOs. It's a really valuable tool that's come along thanks to advances in science, and we're really excited about that, but there's other options out there. We talked about in the science conference how in the rare disease space there's sometimes a lag, like a therapeutic will come out for cancer. Cancer is a very easy indication to get into because there's a huge patient population, there's a lot of money around it. And then a few years later, we find a way to use it in rare diseases. We get creative, we're scrappy. We have to work with what other people are doing. And so we're now looking at additional modalities in addition to the ASOs to figure out how we can use those in Dup15q. We're looking to add our partners. We've had a number of people who we're working with who are hoping to bring into collaboration with us so that we can help them advance their programs. And then we're also looking in therapeutic testing and models closer to humans. So I talked about cells and mice, but before you get to a clinical trial, you want to put it into a rat or into a monkey, and so we've got things moving, which is really exciting. We're moving forward, and I think that's all you can ever ask for is forward progress is really important. So I'm coming up here as a PhD, and I'm telling you about Dup15q research. I'm talking about the fact that we had the science conference with 200 trained professionals, and at the end of the day, the people who make Dup15q research possible are the people at the tables here. It's you guys. You guys make the research possible. It's through your participation in research studies, your generous support so that we can fund our researchers, your distribution of our information. We tell you guys about studies. If you guys get to tell them to your friends who aren't in the Dup15q space and say, look how exciting this is. We're making progress. This could help my kid. Those types of things are really valuable and important. And it's only through your participation and your efforts that research has any chance of succeeding. So I also want to make sure that you guys are aware that any of the research I was talking about here, we've got trained scientists, but they can only do it because of people like you. If you guys want to call to action, if you want to think about ways to help, you can pull out your phone, you can take a picture of these slides, and you can think about some of the ways that you guys can engage in research I'm not asking you to put on a white coat. I'm not asking you to go to school for 6 years and make not a lot of money to get trained for a PhD. I'm asking you to do things like think about your personal network. Do you have people that you know in science that perhaps would be valuable for us at the Alliance to hear about? Can you connect them with us? Can you reach out to the science committee? Can you reach out to me? My email, you get emails from me all the time. You guys can reach out to me and say, hey, what about this? I can tell you if we've done it. I can tell you if we're going to do it. I can tell you if it's worthwhile to study, which oftentimes it is. It's just a question of do we have the bodies to do it? Enrolling in Dup15q research studies is so, so important. And at the conference is the most valuable time because you guys are spread out all over the world. And so to have you in one room is really, really valuable. We also want to make sure that your friends, everyone in this room, you're part of the Dup15q Alliance, you're registered, but there's some people in Facebook groups that aren't part of the Alliance. We want to make sure you encourage your friends to register so that we know who they are so we can support them in meaningful ways. We want you guys to stay informed of the science. One of my jobs is to communicate. I'm here to talk to you about the really complicated things going on in the science meeting and make sure that you guys understand what does this mean for me and what does it mean for the future. So what I'm working to do is take some of those publications that are coming out, we're getting more and more, and giving you guys the key highlights. What does this mean for you guys? What are the key things that you want to pull out of this? And the last thing that I want to highlight here, which has been mentioned briefly, is that we've got galas and walks that go on around the country, and these come from just families. They say, I want to raise some money, and families raise so much money through this, especially if you're in a place where there's a lot of families and there's a need. You guys can raise a lot of money through sponsoring a walk or sponsoring a gala to raise money. And if you feel so inclined to contribute that money towards the science projects, I mean, I would love that, but I know at the Alliance, that's really how we can make some meaningful change. So I just want to thank you all for your attention. I want to thank you guys for coming to the conference and participating in some of our research studies. And I hope you'll take the opportunity to take advantage of the fact that you guys are all really, really smart. You may not have a PhD, but you guys know way more than you're aware of. And so what my job is to tell you what's going on the science side, and you guys can use all of your immense talents and skills and efforts to advance our science in a way that individually the Alliance could never do. So thank you all for your attention. I encourage you to keep our science in mind.